Hello and welcome to the fourth talk in the International Science Month, which is organized jointly by the Science Club and the Campus Radio of ISA Kolkata. It's an international science conference for by students. And today we have a very special guest who would be introduced by a first-year student in our institute, Hannah Nuwais. So over to you, Hannah.
Thank you, Mr. Smith. Yesterday was a dream. Tomorrow is the vision. But a day well lived makes every yesterday a dream of happiness and every tomorrow a vision of hope. Carrying the same vision, we, ISA Kolkata Campus Radio and Science Club family successfully completed 3 talks of Science Month. Good evening, each and everyone present here. Welcome to the 4th talk of Science Month. We extend a very warm welcome to our respected speaker, Mr. Eugenio Contreras, who is a PhD candidate. He did his bachelor in biology at National Autonomous University of Mexico. Then he started his master degree in the same university, focusing on study of the mechanism of regulation of the immune system. In 2019, he started his PhD studying the mechanisms working with biology of Treg. He has been awarded with the Coca-Cola Foundation Fellowship and National Council of Science and Technology of Mexico Fellowship. He has taken classes at different universities such as the University of California, Los Angeles campus, and the University of Minho, Braga, Portugal. He has presented his work in national and international conferences. Mr. Eugenio is very passionate about science and the science communication. He firmly believes that science should be free, open, and shared in an egalitarian manner. I know you all are eagerly waiting to start this astounding talk, so I would not like to consume your precious time. With immense pleasure and gratitude on behalf of the ISA Kolkata Campus Radio and Science Club, I cordially invite Mr. Eugenio to present the session, and I humbly request the audience to post your questions. Now, Mr. Eugenio, over to you.
Thank you very much for the kind introduction, and thank you very much for the invitation. I know it's 8:30 now in India, and so thank you very much for joining. So, do you guys see my screen?
Yeah, we can see your screen.
So thank you very much. As the introduction, my name is Eugenio, and I'm going to be talking a little bit of the work I have been doing during my master's and my PhD under the guidance of Dr. Paula Licona Limon at UNAM. And so if you have any question, please don't hesitate on interrupt me at any time of the presentation so you could get more clear the message of my work. So I work with the immune system, and as all we know, the immune system is the system that allows to detect what is strange from what is from our own tissue. So we need to require a balance for controlling homeostasis in our body, and the immune system has been divided by immunologists in 2 big groups: the innate immune system and the adaptive immune system. The innate immune system includes cells that will protect in a really fierce barriers, like in the skin, in the intestine, in the lungs. So these are cells that are located in those tissues, and are the sentinels, the first cells that will encounter the pathogen. Then later, days after the infection or the pathogen, you will develop an adaptive immune response. And this is basically done by 2 types of cells, B cells and T cells. And together, these 2 cells will induce the generation of antibodies and the production of cytokines that will protect the tissue and the body for future infection. So basically, this adaptive immune system actually have the potential to develop memory and will protect the body or in the individual after a second infection or challenge. And among different things that the immune system actually protects us, one of them is tolerance. And the tolerance is really important because we don't require the cells the immune cells to activate against our own body. So developing a tolerogenic state for protection of our own tissues. Also, we require also the immune system for tissue repair upon damage. And finally, and most importantly, is the clearance of pathogens. And my talk will be focused mostly on T cells, in particular a subset that is called T regulatory cells, that I'm going to talk a little bit more in the next slides. So we now know that during immune activation, we require tolerance and inflammation for control and maintaining of homeostasis. So if we look like in a balance, tolerance and inflammation, we require inflammation for the clearance of pathogens that are in the body, but also we need tolerance for maintaining the homeostasis. And as I mentioned before, tolerance is important for suppress immune cells not to activate against our own tissues or our own antigens. But if we break the balance between tolerance and inflammation, so we can be in a state where an excess of inflammation happens upon autoimmunity, where our own bodies, our own immune cells are actually destroying our tissues, such as diabetes, for example, type 1 diabetes, where these cells are actually destroying beta pancreatic cells. On the other hand, an excess of tolerance happens in cases such as cancer, where the cancer develop an immunosuppressed environment that will actually affect the activation of immune cells for tumor clearance. So hold on. Okay. So one of the cells that are really important in controlling the tolerance and inflammation are cells of the adaptive immune response that are called T regulatory cells. These T regulatory cells, or Treg cells, are characterized by the expression of a transcription factor that is called FOXP3, and we can detect them by flow cytometry by the expression of a receptor that is called CD25, or the alpha change of the IL-2 cytokine receptor. So these Tregs are really important for controlling tolerance and inflammation. And why are these cells actually important? Well, we know that FOXP3, as I mentioned before, is the master transcription factor of these T cells. And what basically FOXP3, the transcription factor, does will give a transcriptional identity to the T cell will activate and repress genes to give the cell this immunosuppressed phenotype. This gene is quite important because people that have worked with this and have developed knockout animals for this protein, they find out that the animal actually died 3 to 5 weeks after birth. And this is because the animals develop autoimmunity. You have a multi-organ failure. All of the T cells are actually activated, and the animal basically dies because of constant inflammation. This is basically because the animal lacks T regulatory cells. So not having these T regulatory cells will end up with autoimmunity and death. In humans, patients with the mutations in the FOXP3 genes will develop a syndrome that is called HIPEX, which is immunodysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. And this basically, as mentioned before, the only treatment for these patients is a bone marrow transfer from a healthy bone marrow without these mutations to somehow now the individual have developed now correct Tregs, and this is also have to mention that it's happening only in males because it's an X-linked syndrome. So only as we all know, the male is hemizygous for the X-link for the X chromosome. So if you have mutation of the X chromosome, you will develop this disease. So basically, and this is I'm telling you this because not having Tregs, and particularly not having the correct expression of FOXP3, will lead to autoimmunity and disease. And one of the signals that are crucial for the generation of T regulatory cells and the correct expression of FOXP3 is TGF-beta. So just before we start talking about TGF-beta, I just want to mention that these T regulatory cells are developed in two anatomical tissues. The first one is the thymus, and these T regulatory cells that are called NT-reg or natural regulatory cells are generated naturally and constantly in the thymus, and these cells will migrate towards the periphery, towards different organs to protect and to suppress clones of T cells that might be autoreactive for several autoantigens. So these T cells, Tregs, will actually protect us from naturally occurring autoreactive T cells. In the other hand, we have Tregs that are generated in the periphery. And when I mention periphery, I mean the mucosas or the joint or different lymph nodes where the human body will, or the body encounter antigens that are foreign to the individual, such as microbiota, or it can also be the diet. So you develop Tregs that will somehow tolerate the microbiota and the diet for not having activation of T cells and maybe inflammation. So in this case, the Tregs are protecting from the periphery in the inside with the anti-reg compartment. So that's what basically these Tregs are really quite essential for maintaining homeostasis of the individual, and this is one of the signals that is protecting us is the TGF-beta. So what is the TGF-beta signaling pathway and how does it work? Well, TGF-beta is a really important cytokine during development and in several cells and tissues. But TGF-beta will signal binding to the type 2 receptor, which is an homodimer. When this happens, now will allow to bind to two pairs of type 1 receptor, and you will find this heterotetramer. When you have this heterotetramer, you will now activate the kinase domain, intracellular kinase domain of the receptor, and will phosphorylate several intracellular proteins. One of those proteins are called SMAD2 and SMAD3. These proteins, now phosphorylated, can bind to a classical molecule that this is the first, when they described the signaling pathway, they found out that SMAD4 was the binding of these phosphorylated proteins, and when binding to SMAD4, they will translocate to the nucleus and regulate gene expression. This is what basically, for many years, this was like the canonical signaling pathway, and everyone were talking about this. There are several studies here in cancer and diabetes. Everything is about the SMAD4. However, new data indicates that there's another ligand to SMAD2 and SMAD3 phosphorylated, which is TIF1 gamma. And TIF1 gamma actually can regulate gene expression in a different manner than SMAD4. So this is basically now what we know of the TGF-beta signaling pathway. And regarding our Tregs, we know that TGF-beta controls many functions of TGF-beta signaling pathway controls different functions of Tregs, including differentiation, function, stability, and proliferation. So how does people have studied this signaling pathway? As I mentioned before, the signal is crucial for development, and so if we develop a knockout animal, the animal will not live because it will die during development. So that's why people that want to study this signaling pathway needs to move to a different genetical approach. And I don't know, guys, if you know the Krylov system, where you can actually deplete a gene within a specific tissue or a specific cell type. So the way that the Krylov system works is, well, you have an animal which have the gene of interest will, in this case it will be J, that will be flanked by two sequences. These two sequences can be recognized by a recombinase, a protein that is a recombinase, and this recombinase, when they found these log sites, will activate a process of recombination and will eliminate all of the sequences that are within the log P sites. So in this case, if we now manipulate the expression of the Cre recombinase to a particular tissue or a particular cell type, then we will eliminate our gene of interest only in the cell that we want or the tissue that we want. So in here, what we are modifying is the promoter. So in this case, if all my T cells express CD4, if I put CD4 Cre, then I will have a system where I can eliminate my protein of interest only on T cells. So knowing that, people have worked studying how is the signaling pathway controlling T regulatory cells in a conditional depletion system where we eliminate molecules of the TGF-beta signaling pathway only on T cells using the CD3 system. So people know that if they eliminate the cytokine, if they eliminate the receptors, the type 2 receptor, the type 1 receptor, or the SMAD2 or the SMAD3 proteins, the animal will develop autoimmunity. The animal they have lack of Tregs, they have lack of differentiation, function, proliferation, and stability. So with this data, it's really clear that the TGF-beta signaling pathway is important for generation of Tregs. So knowing this, the next obvious question, well, if we eliminate SMAD4, then we will recapitulate all of these phenotypes because we see that the signaling is really quite important for generation of Tregs. And if we eliminate the most important protein of this signaling pathway, then we will recapitulate the phenotype. However, this is not happening if we eliminate SMAD4. We have actually Tregs, we have differentiation of the Tregs function. They are functioning well, they have proliferation capacity is correct. And also, the animal does not develop autoimmunity, the animal does not die. They only see a phenotype of stability when there's quite inflammation, but the animal is okay. So with this knowing, my mentor asked if the signal, we need the signaling pathway, the TGF-beta signaling pathway for the development of Tregs. It is not SMAD4, then it has to be another molecule, and then we thought about TIF1 gamma. And this is basically my project, knowing if TIF1 gamma is actually the molecule essential for all of this process. So this is my research question. So does TIF1 gamma is important for Treg differentiation? And we use now this model, as I mentioned before. We use conditional depletion animal where we deplete TIF1 gamma only on T cells. And we also have a FOXP3 reported animal where we can track the cells by the expression of GFP. So I could actually isolate these cells by flow cytometry. So sadly, when we got the animal, we found out that the Tregs are okay. So the animal is actually happy. There's no inflammation. Everything is okay with the animal at first sight. So we were, so the animal was really happy, but we were really sad because we didn't find any sort of a difference regarding the frequency of Tregs. So the animals have Tregs. So next we thought, well, we know that some of the molecules that are regulating the function of Tregs is FOXP3 and CD25, which, as I mentioned at the beginning, is the alpha change of the IL-2 receptor, and it's really important for Treg function. So we thought, well, maybe we're having Tregs, but maybe these molecules, the expression of these molecules might be affected. And so we all did this measure by flow cytometry, where we in flow cytometry, you can not only isolate and analyze the cell population in a given tissue, but you can also evaluate the expression of a given protein within each cell. So when we analyze the expression of FOXP3, the master transcription factor, we didn't see any difference at all. But surprisingly, we see an increased expression of the alpha change, of CD25, sorry, on these T cells. So it was happening something there in the Tregs that they express more CD25. So we are not lost at the with this data. So this is all in vivo. So we took the animals, 8 weeks animal, we sacrificed them, we analyzed the Tregs, and they were okay. They were okay. So next, we thought, what's going on if we took this process now in vitro? So we isolate T cells, naive T cells, and then we activated the T cells. We add TGF-beta, and we look for the Treg differentiation process. So this is how it works. So we have our knockout animal and our wild type animal. We isolate a spleen and several immune organs where we can have naive T cells. We sort the cells and we culture them for 5 days by giving signals of activation, TGF-beta and IL-2. And then by flow cytometry, we analyze the cell populations. And what we analyze is this double positive population, the FOXP3 and the CD25. And surprisingly, now in vitro, we see a difference that T cells that lack TIF1 gamma, we see that they differentiate less. So somehow they have a less differentiated potential. Once again, we see this phenotype where there's no difference in the expression of FOXP3, but we see now a difference in expression of CD25, something that is recapitulating in the in vivo data. So here's the only difference, that now in vitro, we see a less differentiation potential of these T cells. So as you may all know, with this system, we eliminate TIF1 gamma, we eliminate the gene at the moment of the expression of CD4. So this is basically happening on the thymus. When the T cells is generating, all of the T cells are eliminating TIF1 gamma. So we might think that the phenotype that we're seeing, at least in vitro, is because you come from a pool of T cells that from the beginning of their differentiation, they lack TIF1 gamma at the thymus. So we move to a different model where we take TIF1 gamma now only at our in vitro in our in vitro experiments. And so how do we do that? So we move to a different type of CRE model where now our CRE is not bound to CD4, but is bound to estrogen receptor. And it's basically the same model, but now when you add tamoxifen to the cells, you can control the moment where the process of eliminating the gene is actually happening. So what we do is we took the T cells from here, we put it in a cell culture, and now at the beginning of our cell culture, now we add tamoxifen and we eliminate the TIF1 gamma from our T cells. And once again, we look for the expression of FOXP3 and CD25. So just before we start, I just want to have this image. So how does this system actually works? So we have here our gene of interest blocked by these LOXP sites, and we have our CRE protein together fusionated with the estrogen receptor. And the nature of the estrogen receptor is that the estrogen receptor is most of the time, or all of the time, the receptor is in the cytoplasm, but only when it's bound by the tamoxifen or the ligand, they will induce translocation to the nucleus. So in this way, you have a cell that has the LOXP sites, and you can actually deplete the genes that you want, but only you control the timing of the depletion. So this is really interesting because now you can control the time maybe in vitro or in vivo, in which time point you want to your gene to be eliminated. So this is another way to demonstrate our data. So this is, I only have, so as I mentioned before, we have now this animal with the CRE recombinase bound to the estrogen receptor. And I put the tamoxifen at the beginning of the cell culture. And this is, I start this experiment during at the beginning of the pandemic, so I don't have much replicate data, but I have been replicated this experiment only twice, but I see the same phenotype. So when I put the tamoxifen and I eliminate the gene at the beginning of my cell culture, then I see a reduction of the frequency of Tregs and also of the numbers of the Tregs. Surprisingly, we see the same phenotype of CD25 of the IL-2 receptor. We see an enrichment of the receptor when we don't have TIF1 gamma. And interestingly here, we are not sure what's happening here, but with this experimental procedure, we see now that the FOXP3 is actually reduced. So it seems that, yes, at least in vitro, with these two animal models and genetical approach, we see a reduction of Treg differentiation in the absence of TIF1 gamma. So yes, so the next question is, okay, I think this is ground. So our next question was if TIF is okay. So in vivo, it seems that it's not have any function on the differentiation. In vitro, yes, but we thought, well, what's happening with their suppressive function if they're actually functioning well? So we use in vitro suppression model where we test the suppressive capacity of the drugs. And how do we do that? So basically, what we do, we took we isolate Tregs from this animal, and from another animal that had another congenital mark, we isolate T cells, and then we co-culture. If these T cells are actually activated, they will proliferate. And these T cells are stained by an intracellular staining molecule that will indicate you if these T cells are proliferating, the dye mark will be dividing in each cells, and you will see it like in a graph. And I will show you here how does this work. But basically, if you put these T cells with these Tregs, you will somehow test the functionality of these Tregs. So how does this model work? So you start with the T cell populations that are staying with this tag violet, and if every time the cell divides, the dye will dilute. Okay, so we see here in this graph. So at this point, this is the most stained cell, and each peak will indicate that this is come from a cell that divides. So this is, in this case, these one are the cells that more divide. Okay, so in the same experiment, if you put now Tregs, then the Tregs will suppress the proliferation. So if you put more Tregs or less Tregs, then the suppression capacity, the peaks of the division will be reduced. Okay, so knowing this, we tested the suppression capacity of our Tregs that lack TIF1 gamma. So in this case, we put a different ratio Treg with effector T cells. And we see, for example, if we put one Treg with one effector cells, there's a really suppressive ability of our wild-type T cells. And also it's happening with our knockout T cells, so it seems that it's not quite different. But when we change the ratio of effector with Treg, we see that, for example, here, we see that Tregs that lack TIF1 gamma are actually not suppressing as well as the wild-type animal. So yes, I just I have a graph of different experiments where we see that it seems, I don't have the statistics, but it seems that not having TIF1 gamma or Tregs will affect the suppression capacity, at least in vitro. And we also have some data that it might be because of Tregs that lack TIP1 gamma does not have CD73, which is a really important suppressive molecule for Tregs. So we have a hint of what's going on here. This is in vitro. Then we move in vivo with a tumor model where we have, once again, this animal, and we put melanoma. So Tregs are actually bad cells for tumor. So if the Tregs are actually working, the tumor volume will be bigger. If the T cells are not working well, if the T regulatory cells are not working well, then the tumor volume will be reduced. So in this case, we're testing if not having TIP1 gamma will affect the suppressive capacity, at least in a model of melanoma. And this is one data I have where the tumor from the wild-type animal, the ones that have TIP1 gamma, is actually bigger, and the tumor of the animals that lack TIP1 gamma in the T cells, they're actually smaller. So this is a first hint that something is happening with the Treg suppressive functions. So I just want to mention that this animal is actually depleting TIP1 gamma in all of T cells, in all of the T cells of the animal. So now we are moving on to a more specific genetic model where now we are only eliminating TIP1 gamma in the Tregs only. So we hope to see that when we eliminate TIP1 gamma only in the Tregs, we will see the same phenotype. So how is TIP1 gamma controlling Treg function? So just to summarize, we see that at least in vivo, the Tregs are actually differentiated. In vitro, we see affection in the differentiation, but we also see affection in the suppressive capacity in vivo and in vitro. So when we look at the function of TIP1 gamma is or transcription intermediary factor 1 gamma, or TRIM33. It's a protein that has different domains in the amino carboxy in their amino domain. They have this ubiquitin ligase domain that will induce the binding to several proteins. We have a coil domain that will induce the interaction with now different proteins, the SMAD binding domain. And most interestingly, in the amino terminal, we have this PHC and bromo domain, which these domains are domains that allow the protein to bind to epigenetic marks. And as we all know, the PHC or plant homeodomain is a domain that will bind to methylated histones. And more interestingly, the H3K9 trimethylated mark, which is a mark of suppression. In the other hand, you have a bromo domain that will bind to acetylated histones, and these histones, this mark is associated with cell activation. So we have a protein that actually can bind not only activated histone marks, but also repressive histone marks, which make protein a really chromatin reader and chromatin reader of particularly poised chromatin states. So, you may ask why I'm telling you all of this. Why is this relevant regarding the Treg and particularly FOXP3 gene expression? Well, when we look at the FOXP3 gene, we can find several regions. We have the promoters, and we have the exons, and we have these CNS regions, which are non-codifying sequences that are mostly regulatory, like enhancers as well. Particularly CNC2, it has been recognized to be an enhancer that give stability of FOXP3 expression. And particularly Tregs or T cells, we can find these marks that TIP1 gamma can actually recognize. So our hypothesis is that TIP1 gamma is actually binding to this TNC2, particularly recognizing these marks and controlling Treg function. So what do we know about this? At least in our experimental setup, we know that when we do like chromatin immunoprecipitation assay to trying to look at the location of TIP1 gamma in the locus of the FOXP3, we found that particularly TIP1 gamma is enriched in the promoter and the CNC2 region. And this is particularly interesting because some data indicate that these two regions are actually crucial for function and suppression capacity of Tregs. And our current hypothesis is that what is actually happening is that TIP1 gamma is forming a loop between promoter and CNC2 region only during inflammatory conditions. In resting Treg, where there's no inflammation, when there is no TCR signaling, when there is no, the homeostasis, nothing's happening. But when we see a process of inflammation, example, our in vitro experiment where when we activate with TCR, or in our tumor model, when we have inflammation, so now we have activation, and TIP1 gamma might be forming here a loop. And this is just a current hypothesis. We need to test if this loop is actually happening during activating conditions. So this is really a preliminary conclusion. So we know now that TIP1 gamma negatively regulates CD25 expression in vivo and in vitro. TIP1 gamma regulates differentiation of Tregs in vitro. And we know that now that TIP1 gamma regulates Treg suppression capacity in vitro and in vivo. And I didn't show any data, but we have some data that actually indicates that TIP1 gamma is essential for regulation of the stability of Tregs only on inflammatory conditions. And finally, TIP1 gamma localized at the CNC2 promoter of the FOXP3 locus, and we think that it's controlling and regulating Treg function. So thank you very much for your time, and I hope you have I could be clear with my data, and if you have any question, please don't hesitate on asking. Thank you very much, and thank you, the ISER Colcat Science Group, for inviting me. Thank you very much.
Yeah, thank you so very much, Eugenio, for this great presentation, and we really appreciate it. So we have some questions, and the first question is from Tanishta, and they are asking, can there be any other treatment for IPEX, as bone marrow transplant can be a bit of
No, you can treat. What I know, you can treat the patient with anti-inflammatory drugs, but at the end, it will end up with really bad ending, because you might develop cancer. So the only treatment known, as I know, is the bone marrow transfer that will somehow cure the disease totally.
Great. So, so there is another question, and the question is that when we are talking about TF1 gamma and SMAD4, these two proteins with the FOXP3, but we see that even after we stop the function of these proteins, there is still some FOXP3 activity. So, is there any other pathway which is happening here?
Yes. So, it seems clear that the TGF-beta signaling pathway is crucial for the Treg function, but neither SMAD4 or TIP1 gamma. What we know now, and I will go back to my presentation, is that actually we know that TIF and SMAD4 actually can work together for the correct expression of different genes. So we have this hypothesis that a double knockout of TIP1 gamma and SMAD4 will end up with not having Treg or FOXP3. So it seems that you have TIP1 gamma and SMAD4 working together for the correct expression of FOXP3. We don't have the animal, but we think it could be really, really a good option. If not, there are, as mentioned before, there are several proteins that are phosphorylated by the intracellular domain. It is not only SMAD4, so we have signal pathway, we have different proteins here phosphorylated, so it might be a combination of not only the SMAD but also other type of proteins.
Okay, I see. We have another question, and this time by Shantan, and they're asking that it's a big question. We observe that homeostatic proliferation of Treg in vivo can be markedly enhanced by treatment of mice with anti-CTLA-4. The enhanced proliferation of Treg in this model was accompanied by enhanced proliferation of memory phenotypes. Memory phenotypes consistent with a loss of Treg suppressor function. But can you please elaborate the exact function of CTLA-4 in Treg function?
Okay. The enhanced proliferation of the... okay, yes. What basically CTLA-4... So I didn't show a little bit how those Tregs actually function, but Treg can suppress T cells in different ways. So Tregs can, for instance, they can secrete cytokines, like suppressive cytokines, to suppress T cell activation. On the other hand, Tregs can express in their membrane this molecule that is called CTLA-4. And CTLA-4 is a suppressive protein that will bind directly to CD28. So in order to, okay, so I will move to Google to find a cool, are you guys seeing my my screen? My Google image?
Oh, no, we can only see the presentation.
Okay. Okay, so I got a cool image here. Okay, so you have here.
Yeah.
So,
okay.
Yeah. Okay. So in order to T cell to be activated, we required different signals. So for instance, we required TCR signaling. A second signal would be CD28. No? So we have the T cell that is activated by an antigen presenting cell. In the other hand, so these signals, the TCR is a positive signal as well as CD28 is a positive signal. But now Tregs express CTLA-4, and CTLA-4 is a negative signal that will induce the turn off of the activation of the T cells. What Treg actually does, they express CTLA-4, but they express to bind to CD80 or CD86, which is a ligand for the activation of the T cells. So somehow the T cells, the Tregs that express CTLA-4, they're sequestering the CD80 or CD86, not allowing other T cells to be correctly activated. And if the Treg and the T cells are not correctly activated, they will go to anergy and they will not be activated. So the main function of the main function of CTLA-4 on Treg is basically to compete with other T cells for this the ligand of this of CD80 or CD86 for T cell activation. So going back to your question, you say that the treatment with anti-CTLA-4, the enhanced proliferation in this Treg model, it will end up enhancing proliferation because you are blocking CTLA-4 expression from Tregs. So now the T cells, whether a CD4 or a CD8, can bind to CD80 or CD86 to be correctly activated and proliferate and generate a T cell memory.
Great. That was a nice answer. And I think, Sayantan, if you have any more doubts, then you can write away in the chat box or contact Eugenio by email if you want to. I think he'll be glad to answer your question. The next question is, what are the clinical implications of your study or your research?
Sure. Yeah. So what we want, as we all people that do basic science, is to move toward more clinical implications. So what we want, and our lab is focusing now, is controlling the function of Treg by modifying the TGF-beta signaling pathway. So we could give inhibitors of the signaling for controlling the Tregs. And now we can look for inhibitors of TIF1 gamma or inhibitors of the PHC or bromodomain to somehow controlling the Treg function, particularly on the tumor microenvironment. Or on the other hand, we can potentiate the Treg function to control autoimmunity with this, maybe by purifying Tregs from a type 1 diabetic diabetic patients and potentiate the Treg function by knowing all of this signaling that is controlling their function. So basically, that will be the ultimate goal. And now we are at the in my lab. In my lab, we are focusing not only in the TIF1 gamma on Tregs, but also on CD4 or CD8 T cells. So somehow try to dissect particularly what is going on with the TIF1 gamma in the tumor microenvironment.
Great. So the next question is the in vivo models or the rats that you remove the TIF1 or knock out the TIF1 gamma. Do they go on to develop any autoimmune disease or any sort of disease or in their lifetime?
Yeah, that's a really good question, and that's the very first thing that people look. If you have a molecule that is important in Treg function, the animal will develop autoimmunity. We haven't seen any type of autoimmunity. However, we let the animals to be old, so we expected to see. Okay, so in the elderly, you have a constant chronic activation, constant chronic inflammation, and we see that these animals, when they are old, they have enhanced. They are like more You see more inflammation on these animals, and also when we look at Treg markers of activation, these Tregs are actually not being well activated. So there's a problem there. They are all of the time they are resting. It seems that there's no good activation of these Tregs. So that's the only phenotype we have seen. Now we are moving to, as I mentioned before, to this genetic approach to only eliminate TIF in the FOXP3 pool, and we expected to see the same phenotype.
Yeah, that's really interesting. And Pratya is asking a question, and the question is, what was your inspiration behind becoming an immunology researcher?
At the beginning of my career, I wanted to study biology because I was good at it, and I really like to see to nature. And at the beginning, I wanted to work with plants. So I wanted to work like in a forest, and that was like my first goal at the beginning. But at the beginning of my the first years of my career, we see all of this molecular biology stuff and microbiology, and I was really interested in how does the immune system actually protect us. And every time I ask like question about immunology, people ask, "Oh, it's really difficult, it's really hard." So I started reading, and I really started passionate about it. So that's why I really, and then I wanted to go to cancer biology. In fact, people in my university called me the cancer boy because all the time I was talking about cancer. And then it was when I when I went to UCLA, when I took this immunology class, and I was really fascinated with immunology. And then I came back to Mexico, and I decided I wanted to become an immunologist. So that's why.
Yeah, that's really interesting, and that's cool. So we'll take one last question because we're running out of time, and the question is, how has the research and what sort of tips would you want to give to researchers or students who are trying to work in this pandemic?
Yeah, so it really had for me. It had really been hard experience because, as mentioned before, I have tons of animals. I have like 8 to 9 strains of different animals, which I have to take care of them, and everything is closed, and I need to go to the lab to look for my animals. So you have all this worry about to become to contract the disease, to get infected. So you have like this anxiety of have to go, and then you also have this other anxiety of people of I have to finish my PhD in 4 years, then my scholarship will end, and so you somehow become anxiety. However, so you can see the pandemic in two ways, the bad way, or you can also, and I always try to be positive. Sometimes it's hard, but I try to be like to look in the other way, see what's bringing positive the pandemic. And so I think with this pandemic now, and more opportunities for students from different parts of the world, particularly developing countries such as Mexico, that we don't have really access to good science or good conference. It's all everything is in Europe or in the USA or China. And now with this pandemic, you have the opportunity to have access to all this cool science that is done, and it's all free. If you go to Twitter, you will see that there's a lot of science conference, really high quality there. And this brought me the opportunity to not only to get access to this science, but to be part of that. So as I don't know if you know, but I created. I had the opportunity to found this public journal club where we discuss articles with the ones who made the science. So with this, it brought me a lot of opportunities to interact and network with scientists from all over the world, and to see that we are. I mean, at least in Mexico, we are really passionate, but we think that we don't have enough money. People in the outside are really is better because they are actually doing better science, but they're doing better science because they have the money, but we have no difference between each other. So that made me really grateful, and to think that I could be I could follow this pathway, because I think I really I have the tools for that. I have I have the tools for becoming a great scientist, and I have also the passion. And I think it's something that you need to find, to have the passion of what you're doing. Don't feel anxiety for everything that is going on. And I will really highly recommend you to become professional on programming, bioinformatic tools. I think they're really, really good tools for. That's the only thing that is, at least for me, is missing. I need to go on that pathway. But if you are passionate, then you will have no limits. So yes, I hope you find a message in what I'm saying, and please don't quit on science, and keep reading. There's a lot of opportunities there, but they are there. You just have to join in to learn a little bit more.
Thank you so much, Eugenio, for this really inspirational talk. And so that was all for today. And we thank you, the audience, to come here, and we thank Eugenio to take time off his busy schedule and give wonderful talk. And it was really interesting, and also this inspiration. So we'll be stopping the stream now, and whoever's watching, we'll have future talks announced, and it will be sent to you via the Institute webmail. And so we'll see you in the next stream. Thank you so much for being here with us, and thank you, Eugenio, for being such a wonderful speaker. See you all.
Thank you all for your time.